Hi, Patricia. This is Paula.
Hi, Paula.
How's it going?
It's going good.
Good. I'll just give it a few minutes for everyone to join in. So I'll introduce the two of you and then hand it off for you guys to get going.
Okay.
Can you just confirm, Lauren, is your last name pronounced Stainback?
Yes, ma'am.
Okay, perfect.
Honestly, I don't know if that's really how it's pronounced. It's just how we've
Okay,
pronounced it for a couple generations now. So that's,
okay, that works.
That's where it landed.
I'm not particular.
Perfect. Okay.
Patty, that's a beautiful image. I meant to tell you that earlier.
Very colorful,
Hmm.
just like our logo.
Good morning and welcome, everyone. Thank you for joining us. We are recording the presentation today, so we can send this out along with the slides to everyone afterwards. If you have any questions, please put them in the chat box and we will answer them at the end. So I'd like to introduce our speakers today. First, Patricia Atkins is the Global Product Manager for Specialty Chemicals at Specs NSI HPS Chiron, where she has been a scientist for the past 15 years. She is a graduate of Rutgers University and has been involved with many industry-focused advisory organizations and is a frequent presenter at conferences. Patricia is a published author with her work appearing in journals and trade publications, including spectroscopy, LCGC, and Cannabis Science and Technology, where she's a columnist for analytical issues in cannabis testing. And then Lauren Stainback is the Global Product Manager for Microbiology and PTs at Specs NSI HPS Chiron. Lauren leads the microbiology product and business development efforts. Formerly a forensic lab director, Lauren has 15 years of experience within clinical and analytical laboratory science. Lauren is focused on supporting customers and distributors through technical support, applications training, and new product development. So I will pass it over to you two. Thank you.
Thank you very much. So we're going to be talking about some of the best practices for food and beverage testing for food safety. In the US, foodborne illness, there are 48 million cases of foodborne illness a year. That means 1 in 6 people will come down with a foodborne illness, and that results in about 3,000 deaths a year. Worldwide, the World Health Organization said there are 300 million foodborne illnesses, with 1 in 10 deaths, and there are 420 deaths yearly worldwide. Now, all of these deaths and illness come down to about 250 potential sources, and they divide up into physical, chemical, parasites, and microbiological. And these can be from contamination issues from poor sanitation, poor waste handling, poor food handling. They could be industrial contaminants. They could be agricultural contaminants. So they're all different sources for foodborne and waterborne illness. Now, if we look at the pathogens and potential targets in food, we are talking about everything from microbiological to chemical. Natural toxins. There are natural microbiological organisms. There are natural toxins and natural elements that are present in the soil and in the water. Things like cyanide in different fruit pits. You have adulteration and counterfeiting. Things when they add like lead to make something look whiter or to make a product look in a certain way. Process contamination, like nickel from wear metals or acetone from some type of extraction. And then handling or cleanliness. That's where you get your E. coli and some other microbiological bacterial contamination. And then packaging contamination. You can have heavy metals, antimony, all sorts of other packaging materials, phthalates, inks, dyes. If you look at the life cycle of food contamination, at every step from the initial agricultural farm where the product is first grown, you have contaminations blowing from maybe you're growing an organic farm, but the farm next to you is not organic. So then you have pesticides or contaminated water or contaminated soil, then moving into that farming or that industrial growth. Then you have farming, the farming itself. Does that add contamination to it? Storage, can you get molds and other spores and other parasites? You can have rodent droppings and bird droppings. So there's the potential during storage. During transportation, there is a potential for a product, if it has to be refrigerated or kept at a certain temperature, to become overheated or to get frostbite or the packaging to collapse or break because of improper handling. Then you have the processing of the food to a form that gets sold in a store. Is there process contamination like wear metals? Is it put through like potatoes? So they grind up all the potatoes and they make potato chips out of it. Is there going to be wear metals in that package of potato chips because of the processing? Then you get to the packaging, you have inks, you have dyes, you have sealants, glues, different adhesives. You have possibly coatings inside the package, like some cans have bisphenol A on the inside of the can. So then you have the potential for contamination there. Then through the grocery store and how is it handled? How is it stored? And then finally to how it arrives in our home and how we ourselves treat the product. Do we actually allow for microbiological growth or growth of yeast and mold in a product because we don't handle it properly? So the chemical targets we're going to look at are organic compounds, organometallic compounds, and inorganic compounds. The organic metallic compounds are kind of the bridge between the organic and the inorganic compounds. So what makes a chemical dangerous? Well, how much of that chemical are you exposed to? Are you exposed to a lot or a little of that chemical? What's the form of the chemical? Is it one of the chemicals that actually has a lock and key mechanism in the human body, like an endocrine receptor? So if it has this kind of functionality that allows a lock and key effect, then it is potentially more dangerous than if it can just be flushed through the body. And how long are you exposed to this chemical for? Is it going to be a short term or an acute exposure, or is it going to be a chronic exposure? So the longer and the more of these elements that are involved, it goes from acute exposure to a chronic exposure. Your exposure can then happen in several different ways. You can absorb it. And if it passes through the tissues and in across the blood-brain barrier, that particular chemical or that particular substance would be more dangerous. That one that just would then absorb into your system, then flush out through your kidneys. Distribution, where do these chemicals travel to? As I said, if it just flushes right out of your system, not as dangerous. If it accumulates in a particular body tissue, does it accumulate in the lungs or in the heart or in the brain? And does it transform in the body, or is it subject to bioaccumulation? Can it accumulate? Is it one of the chemicals like vitamin A that can accumulate in tissues, or is it like vitamin C and it flushes right through the body? So there's also roots in food packaging, the structural material, the plastic itself, or the mylar, or the metal, or the paper, any catalysts used to produce those materials, any copolymers or stabilizers, fillers, extenders, inks. Inks are a really big source of potential contamination of food, colorants, plasticizers. As we said, there are a lot of phthalates, there are coating materials, flame retardants, lubricants. There's a lot of slip agents and blow agents when plastic bottles and plastic containers are made. And of course, you have the adulterants as well. You also have processing residues where when you actually make a petite piece of packaging, that there might be residue. We all have had the experience where we pick up something from the store, like a piece of plastic material, and it has a very strong phthalate smell, or it has a powdery substance on it, which we know is processed residue. Unintentional reaction residues. So sometimes they make a reaction to cause a particular food to be produced, or a particular process, like puffing up a particular type of like corn puff or something like that. And maybe there's some residual gas, or there is some residual solvents. Microbial contamination, accidental additions or over additions, unreacted materials, machine residues, and recycling residues. For inorganic compounds, they are the really dangerous ones are the ones that we know. We call them, the toxic metals or the heavy metals: lead, chromium, antimony, arsenic, cadmium, palladium, platinum. These can be very toxic. Then we have the ones that tend to show up as wear metals. Again, we see chromium and lead, nickel, magnesium, copper. These are all that can be part of machinery parts, part of processing. Then we have the parts that we consider to be often catalysts, that they can be used as a catalyst, or they could get used in the processing and can be reused. Then we have inorganics that we consider to be nutrients. The funny thing about all of these is that they can cross over into many different categories. So you can find lead in wear metals, but you can also find lead as a toxic element. Antimony can be part of a catalyst, but it also can be a dangerous toxic component. Chromium can be a wear metal. It can be a dangerous inorganic, but it also can be a nutrient. So it often says the dose is the poison. Well, that is true. It is how much and in what form that particular chemical comes in, if it's dangerous to the body or not. So the inorganic forms tend to be less poisonous than the organic forms, and there are a few exceptions, but in general, the inorganic forms tend to be less poisonous than the organic forms. So methyl mercury is much more toxic than inorganic mercury. The exception are the arsenics. The organic arsenic forms are less toxic than the inorganic arsenic forms. So again, how long and how much are you exposed to? Here are some common elements that you find in packaging. And you can see these are some pretty dangerous elements. You have mercury and lead, like lead chromate. You have antimony. So there's a lot of possible packaging elements that can potentially contaminate your food. And then you have the plastic itself. There's a lot of plastic that has some warning signs. There are things like a PET, which has the potential for having some copolymers like antimony. Antimony is a catalyst for it. You have PVC, you have lead and chloride in that. For polystyrene, you have the styrene compound, which can be very carcinogenic. Polycarbonate, you have bisphenol A. So all of these are normal parts of the manufacture and the processing of these plastics, but these residues could potentially be in contact with the food. And then finally, the PLA, you have pesticides because it's a natural bioplastic. So when you have plastics also, like a plastic soda bottle or a plastic milk container, or some sort of canned food or a plastic bottle with food in it, you also have a leaching of those elements into the actual food. And the longer that it's in contact with it, the higher the amount of inorganics that are going to be transferred into it. And if you have a higher pH, even more because it's going to leach even more metals. Acids will leach the metals from the container. So think about your tomato sauce or your soda. Those are going to leach any potential inorganic contaminants into the tomato sauce or the soda just by their very nature, the acid nature that leaches it. With specs, we actually leach our bottles with acid in order to reduce any potential contamination that might interfere with our standards or CRMs. So we rinse and leach our bottles with acid to make sure that that our standards are not going to pull any residual elements from the packaging we put it in. And what we have found out is that by doing this acid wash, we get greatly reduce any potential carryover from the packaging itself. Our organometallic compounds, now these have become very, very important lately. An organic metallic compound has an inorganic functionality and an organic functionality. And this is why it can be very dangerous because remember that lock and key situation, this whole organic, inorganic means that you have a molecule that that can actually fit into multiple different situations in the human body. And these organometallic compounds are widely used in paints, in additives, in fireproofing or fireproofing foams, anti-stick things like the anti-stick coatings and the coatings like Scotchgard and Teflon and things like that. So here are some organometallic compounds that are used in packaging. And you can see, again, these are a whole host of potentially very dangerous compounds, cadmiums. And of course, the most important one we've been talking about for the last couple of years are the PFAS compounds. Those are the per and polyfluorinated alkyl substances. So you have a fluorine and then you have an organic grouping of it. So if you can see it has a lot of inorganic fluorine and it also then has some organic functionality that can interact with human biological processes. Now the compound itself is used, these compounds are used very widely. Like I said, nonstick coatings, they're used in microwave popping bags. They used to be a PFAS coating in the bags. Pizza boxes, that kind of waxy coating interior, they would use PFAS compounds. Believe it or not, they used it as a leavening agent in bread, which I found to be very shocking. And the molecule itself has the lipophilic end and the hydrophilic end. So it is able to biologically interact in a lot of different systems in the body. And of course, it does that lock and key thing. It does that lock and key thing, which means it increases the distribution and they're very persistent. They call these the forever chemicals. For organic compounds, you have, again, we have to worry about that lock and key. If we have that lock and key and it fits into a biological receptor, then that particular chemical can then replace a hormone or another biologically active component that the body really does need, maybe a vitamin or a cofactor. So if you happen to have, this is your let's say this is your vitamin, the little green triangle has the functionality of your vitamin or your cofactor, and then you have a mimic or like an endocrine receptor. It has something else. Well, it could then actually go in and block it so that you cannot get the correct molecule to where it needs to go. So it will actually block it then. You will not be able to get the correct vitamin or cofactor to the receptor. One of the big categories of these mimics or the endocrine disruptors are the phthalates. These like the DEP, the DEHP, and these make plastic soft and they are part of a lot of different coatings and manufacturing processes and slip agents and container material. So they are very, very widely used. Now, people say, well, it's just packaging. What's going to happen? Well, there is migration in packaging. So you have the material and you have your product. When you have absorption through the surface, you have the material go to the surface, molecules of the material go into the food product through adsorption. And then that means it permeates through the material. This is when you have molecule that permeates through material and then it breaks down. This then becomes soluble in your product and you have a dissolution into smaller particles. Absorption, you have then the liquid. Let's say you have a juice pouch, and part of that juice pouch then gets absorbed into the material. That is absorption, liquid into a solid. And then this whole process is the migration, where you have the migration of chemicals into your product. And this happens over time and can increase with spikes in temperature. So when we make CRMs for food, beverage, and packaging, we're talking about heavy metals, toxic metals, wear metals, plastic catalysts, but we're also talking about nutrient metals, additives, of course, our PFAS compounds and organometallics, plastic standards, pesticides, pollutants, vitamins, additives, and colorants. So these obviously group into our inorganics, our organics, and our organometallics. And we're really happy to announce that Chiron in Norway has joined our company over the last few months, and they are one of the world's finest producer of PFAS compounds and PFAS reference materials. So we will be very happy that they have joined our ranks. And when our stock materials are not available, you can always order a custom material. We have custom standards for all GCMS, LCMS, ICP, ICP-MS. Now I'm going to hand it over to Lauren, and she's going to talk us through our microbiological targets.
Hello, everyone. I'm Lauren Stainback, and thank you, Patty. So everyone's heard about all these food recalls and plants that are getting fined by the FDA because they have microbiological contamination in finished food products that are going out to consumers. They're in grocery stores, on shelves and freezers. So everyone's, either known someone who got food poisoning or may have unfortunately experienced this themselves. So I'm going to talk to you a little bit about how our reference materials are used by laboratories to do surveillance testing, to QA approve raw materials, to get used in production environments, as well as doing routine quality assurance and shelf life matrix spikes, things like that. But let's talk about these risks that are facing the public, and obviously can backfire on the brands that manufacture these products. So we talked about how 1 in 10 people globally have experienced food poisoning, and we have 420,000 deaths as a result of having some sort of contaminant in food causing human tragedy. So we have a classification of different contamination agents here on this slide, and you can see the toll that it takes, the death rate there. Look at Clostridium botulinum. That's botulinum toxin. That's common in some canned food items. When you see a can that's swollen or something, it could potentially have botulism, so you don't eat it. And we have, a U.S. death rate due to the Clostridium botulinum 17.3. That's very, very high. It's very common. And then Listeria is not to be outdone by Clostridium. It's started taking people out at a rate of 15.9. Now, the reason why these things are a concern is because they are naturally present in the environment. They grow alongside the crops that are harvested, that are used in creating the food products themselves or creating the flavorings that go into the foods. So there's a lot of monitoring and testing of raw materials that has to occur, and laboratories need to use reference material, bacterial reference materials or viral reference materials, in order to properly ensure that if the contaminant is present in the raw material or in their facility, they would be able to detect it. So let's go to the next slide. Look at all these toxins here. These are much trickier to detect and quantitate. Any of these being present is a no-go. It means something needs to be stopped, product needs to be recalled, or the raw material itself needs to be rejected, and usually a full cleaning process of the facility has to be undertaken, or the areas in which it was detected, or the manufacturing line in which this toxin was detected needs to be completely broken down, taken apart, and proper decontamination procedures applied, which results in downtime, which results in lost revenue. But lost revenue is a lot better than killing someone. So look at the different fungi and their mycotoxins, and then look at the sources on the right. These molds that cause these mycotoxins and the bacteria that cause these enterotoxins are commonly found in these source materials. But why? Why is that the case? Well, these things have evolved together over millions of years. They live synergistically, and the fungi or the bacteria present with the crop actually helps it absorb nutrients from the air, soil, and water. So they've grown in harmony with one another, but unfortunately in mammals, they cause extreme toxicity. There are some toxins that at a certain dose are fatal, but at less fatal doses produce delirium, psychoactive results, basically getting people sick, out of their minds, or high. You'll see ergot alkaloids, and that's common in cereals and grains, and it actually was a big problem. They think it led to some of the Salem witch trials back in the day. So this has been going on for a long time, but now we're better able to detect things because we have reference materials and we have standardized test methods that the labs can use. Let's go to the next slide. So our bacterial certified reference materials allow labs to take a freeze-dried pellet that contains concentration of bacteria, hydrate that material, basically activating the bacteria so they can reproduce and grow. And then you're going to use that freeze-dried bacterial reference material to approve your media, to show that the media is capable of growing what it's supposed to grow. So if that bacteria were to be present in that raw material, let's just say cornmeal, if you process cornmeal at your facility, or you make corn muffins that go in the frozen food aisle at the grocery store, you have to be sure that if there were a bacterial contaminant like Listeria or a mold like Aspergillus, that you would be able to detect it. So what do? You take an Aspergillus and you spike it into your cornmeal, and you ensure that your media would be capable of growing it, and it would be a positive control. So we sell these positive controls already pre-certified, so you know exactly where your results should be if you follow the steps to hydrate the pellet and inoculate your media. So we're giving you a pre-tested bacterial reference material that you simply hydrate and use. Customers buy our products for media QC testing. They use them as positive controls, or they'll use a non-target bacteria that they're not expecting to be able to grow on the media, to just ensure that the media is not going to grow something it's not supposed to. So those are negative controls. They do matrix-based spike recovery studies. That would be checking to make sure there's not any kind of inhibition going on, to where if a live bacteria were present in the material is not masking the recovery, or it's not, growing exponentially, or it's maybe inhibiting its growth, but it's still present. So if that product sat on a shelf, it would grow out and it would cause a problem and give someone food poisoning. So they do matrix spike recovery studies with these. And then also labs that are ensuring that their analysts and the test methods that they're performing are capable of producing accurate results. They will perform method validation studies using these bacterial reference materials. This is how the reference materials are handled by the laboratory and used. They'll take our freeze-dried pellet, which is vacuum sealed in a glass vial, and it stays stored in the freezer. And they will add that to a suitable hydration fluid, which, like I said, activates the bacteria. It wakes them up. They can start reproducing and growing and resuscitating themselves. And so they'll take that pellet, they'll put it into the appropriate solution. They can optionally perform serial dilutions if they choose to. The analyst would take those serial dilutions and then plate them on the appropriate media, incubate that for the appropriate duration and at the appropriate temperature that's called for by the analytical method. And then after the incubation is complete, they will remove the media and they will count the colonies. So our certified reference materials are used in that way to, like I said, for those various applications such as matrix spike recoveries, method validations, or just QA and QCing of media. Next slide, please, Patty. This is an example of that certificate of analysis. When I'm telling you that the products are pre-tested, pre-certified, what that means is you're buying a pack of 10 or 20 of these vialed pellets. They contain bacteria in that freeze-dried pellet. The certificate of analysis for the reference material is going to provide you with our certified activity, which is the colony forming units per pellet, and the standard deviation of that result. But we're also giving you a guidance of the acceptance limits. So this is what you as the laboratory customer can expect to see when you utilize the products on the media and you're performing a similar type of hydration and plating scheme that we use to certify the product. You're going to fall in the same results as our acceptance limits. So you can use those acceptance limits as a quick guide, a quick and handy guide to where you can expect your recoveries to be. We also talk about these products for 3M Petri films. So a lot of you are utilizing 3M Petri films for food testing because they're very convenient. It's a lot more convenient to have these thin, dehydrated film-based media plates than it is to have thick agar Petri dishes stacked up in your fridges. So it's more economical. It also maximizes the space that you have to store your media. So we have thousands of customers that are using Petri films globally. And we've designed some products specifically for use with Petri films. And these products are sold in packs of 10 and 20. We have a great product demonstration video just to bring the point home of how customers are using the product and the advantages of this type of product. But what this is, it's still a freeze-dried pellet in a vacuum sealed glass vial, but this is multiple species of bacteria blended together. So you can hydrate one pellet and you would be able to QC multiple different 3M Petri films. So these are available through Midland as well, and they're a great way if you're QCing a lot of tests each week, you're using multiple types of Petri films. This is going to save you quite a bit of time and money. And this is a feature product that is only available from, NSI through Midland. So take a look at this product group and discuss it with your micro lab team. If Petri films are something that you're using, this is a really unique product that you'll see a tremendous time and cost savings by utilizing something like this with multiple bacteria in a single pellet. Next slide. This is the example of the certificate of analysis. Again, it's got the same information, but these quantitative CRMs will eliminate guesswork for QC reviewing of your methods. And the biggest reason why these multi-organism CRMs are useful is because food products they never have just one contaminant. They'll have chemical contaminants, they'll have multiple different bacterial contaminants. It's never usually just one thing contaminating a mishandled material or a missed QA step or a missed cleaning step in the production of a finished item. It's usually multiple different, multiple sins were committed. And so you have multiple different types of contamination. With bacteria, that's very true. So this will let you QC multiple different bacterial species in a single prep. And so we've got each analyte certified. We've got each analyte's acceptance limits. We talk about how we arrived at the results. We have used 3 different matrices with the bacterial pellets. We've done the hydration fluid only. We've also done hydration fluid plus a dairy matrix amendment and certified that. And then we've done the hydration fluid plus a meat matrix amendment and we certified that. So whether you're a meat, dairy, or beverage manufacturer, we've got these multi-organism products for your Petri films. Next slide, please. So a lot of the customers that I've met with that are in these large food production environments, they're doing their own cell banks. They're culturing their own materials. And so we have these culture swabs available through Midland that allow you to make your own cell banks or just do streaks on media. So these are kept in the refrigerator. They have a shelf life of 12 to 18 months. It is strain dependent. These culture swabs have sterile hydration fluid at one end and the freeze-dried pellet at the other end. You'll simply snap the valve, squeeze the bulb just like you would a Pasteur pipette. You'll squeeze the fluid out. It will come out through the swab end and hydrate the freeze-dried pellet. And then you will shake it to homogenize it. And then you can withdraw the swab itself, the inoculated swab, and you're able to streak your plates. So you can do 4-way streaks if you want to isolate colonies with this, or you can just streak the entire plate surface and grow a lawn. We have customers that will hydrate the pellet and pour it directly onto a Petri film to QC that, because it is 1 milliliter. Once it's fully, once the hydration fluid is fully squeezed into the other end, it is 1 milliliter of saline. So you can use it for Petri films as well. And these are traceable cultures. They are reference materials. They are available through Midland from the NSI brand. And we have about 85 different species available. Next slide, please. This is a visual of what I just described, of how you would hydrate the pellet and then streak the plate, incubate it, and then examine the growth after incubation. But these are just qualitative products. They are not available quantitatively certified. So they don't have an exact CFU value. They have an approximated CFU value. So they are formulated to deliver several 100,000 CFUs per 100 microliters. But you're going to get about 100 microliters on the swab tip each time you insert it into the fluid and withdraw it. So you could have easily have too numerous to count on your plate as a result of using the snap sticks. Next slide. So our specialty chemical portfolio, now that we have the 4 companies, Specs, NSI, HPS, and Chiron, we have the broadest portfolio of reference materials and certified reference materials. We cover a variety of different instrumentation with our analyte, comprehensive portfolio of analytes and concentrations of those analytes. We have certified them using these different methodologies that incorporate these pieces of equipment and instruments. We have PCR kits for molecular biology work. We also have the live cultures, and we have reference materials for food chemistry that are, you can perform the testing using your ELISA plate readers and your ELISA based tests. So we do have the broadest portfolio, and these are available through Midland Scientific. Thank you so much. Patty and I will be here to answer any of your questions.
Thank you. That was a great presentation. If anyone has any questions, you can go ahead and put those in the chat box now, or you can go ahead and unmute yourself too. That works.
I think we've rendered everyone speechless, Patty.
Yeah, I know. I agree. One of the important points when Lauren was talking about toxins from different fungi and bacteria, there are often 2 ways that are regulated. You can either regulate the organism itself. You can regulate the aspergillus, where the laboratory will have to test for the aspergillus, and it can't be usually like what, 1 colony forming unit.
Yeah, like it depends on the kilograms of the finished material. It'll be like less than 10 CFUs per kilogram, or less than 5 CFUs per gram, something like that. It depends on the nature of the product.
The other way that sometimes a product is regulated by the amount of the chemical aflatoxin or ochratoxin or the other type of chemical toxin that comes from the material, the mycotoxin that comes from the material. So usually it's one or the other. Sometimes they ask for both. So just because that you think that NSI only does the microbiology. That's not true. They also actually
Right.
do the chemistry portion. So you would be able to either through specs or through NSI to get both the microbiology and also get the chemical standard that goes along with the corresponding organisms toxin. So
That's a
yes.
great point, Patty. Great point. Because a lot of times your food item or your raw material might have gone through like irradiation or some type of heat sterilization step, but that doesn't destroy the aflatoxin or mycotoxin or enterotoxin that's present. Those are very persistent. At that point, they're just the chemical aftermath of a contamination due to spores of bacteria or mold. But the toxin's still present. So you would still need to do chemical testing of the raw materials or finished materials to ensure that that toxin, although it's not recovering a live organism, the toxin may still be present.
Well, yeah, because you can even have food products that have very long shelf
Mhm.
lives, where by the time you possibly could have the product, it might be a year or more old, and the organism itself has died off.
Yep.
But the aflatoxin or the ochratoxin, the other mycotoxins that were left behind, they will persist and persist because they are very persistent chemicals. Even heat barely destroys some of them. So
Yep.
there's actually been a lot, especially when it comes to the industries like cannabis and nutraceuticals. There's been a lot of talk. Well, it doesn't have any live aspergillus. It doesn't have any live organisms. And oh well, it just has a little bit of the but some of the most poisonous toxic chemicals on the planet are actually mycotoxins, are
Mhm.
these toxins. Gram for gram, less amount of a mycotoxin can kill you than rattlesnake venom. So yeah, so it's kind of odd to think that the organism might be dead for a year, but
Mhm.
its legacy will live on in the product in the form of the toxin it left behind.
And we've got the reference materials for both. We've got the mold the bacterial cells that produce the toxin, and then we've got, so there are live cultures that are available as reference materials, and then you've got all the individual toxins, and you've got mixes of the toxins and different concentrations of those toxins, so you can build cal curves and things like that much more easily. So we've got all those bases covered. We don't have rattlesnake venom yet, but we got the other stuff.
Any other questions?
Is there anyone that has any questions on how maybe they have some applications and they're struggling to find reference materials and they want some guidance? Feel free to discuss it with your sales rep and they can get in touch with us. And we can see if we can figure out some of our product solutions, or maybe even figure out if there needs to be a custom solution developed. So just let this kind of be a starting point for you to think about where you might have gaps in your QA testing, or where you're having difficulty sourcing a reference material or some controls or standards. Use this. This is a two-way conversation, folks. We're here. We know the products, and you have the need. So we just wanted to make ourselves available to see how we can marry those two together and help you out.
I'm also happy to say that Specs was the very first developer of plastic standards for phthalates. So we have plastic standards that have measured phthalate values for packaging testing. That becomes very important for food packaging, to making sure you're not getting phthalates in
Absolutely.
your juice box or phthalates in your food. So I'm very proud I was involved with the development of the very first plastic standards, and those are also available to you as well. Paula, does any of your group have any questions or comments?
I think we're good here. This was an awesome presentation this morning. Thank you everyone for the time. And like I said, we'll send out the recording along with the slides to everyone who registered. So thank you, everyone.
Thank you. It was a
Thank you.
pleasure. Bye, everyone.
Bye-bye.